Guys, welcome back. So I can wind up and call it a night. And we're simply talking about hybridization and hybrid. We've talked about the monohybrid, what a dihybrid is, what a monohybrid cross is, and what a dihybrid cross is. The other terminologies that we encounter throughout, or rather in our course, is the P, F1, and F2 generations. So P is simply the parental generation. So it's simply the first set of parents that are crossed. So in the case of a crossing between dominant, homozygous dominant and homozygous recessive parent, so we have capital T's and small letters T's, they are being crossed. So this is the parental generation, the first set of parents that are crossed. And then we have the filial generation, or F1. So filial simply means son. So simply means this is a generation that is produced from the crossing of the first parent. So the first parents are going to have offsprings, and this is known as filial 1 or F1. And then we have F2, which is the first generation progeny of hybridization. So hybridization here from the parents we have crossed this homozygous dominant and a homozygous recessive parent producing F1. And then under F1 here we've crossed F1, and what will result is what is known as the F2 generation, or rather, yeah, the F2 generation. So this one is the F1 generation, and then when we cross the F1 generation, what we'll have is the F2 generation. So the progeny of hybridization, when it is hybridized with any of its parents here, what will be produced is F2. Well, F1 is the first generation progeny of hybridization. When we just do the first hybridization from the parents, what will be produced is F1. When we cross parents, the F1, when we cross F1 parents, because this now becomes the parents for F2. So what will be produced is F2. And then we have the crosses also. We have the crosses. So when we look at the crosses, a cross is simply, in genetics, when we talk about a cross, it's talks about a purposeful mating of two individuals, which results in the combination of genetic material in the offspring. So that's a cross, and we've been talking about crossing for a while now. So that's what a cross is. And then we have some types of crosses that exist. We have some types of crosses that exist. So for example, we have a reciprocal cross. So in a reciprocal cross, it means that two reverse crosses in which the sexes of the parents are interchanged is done. So if the traits are autosomal, the reciprocal cross always yields the same results. However, in the case of sex chromosomes, the reciprocal is going to yield a different results. So in a reciprocal cross, as we have said, we are going to reverse, it simply means two reverse crosses. So the sexes of the individual are going to be reversed. So for example, we have a tall individual there. So that sign there, it's for a male. That's a sign for male. And then we have a dwarf female. So we have a tall male being crossed with a dwarf female. And then these sexes are reversed, they are interchanged. we have a dwarf female now being crossed with a tall. We have a dwarf male rather being crossed with a tall female. So if the traits are autosomal, what we are going to have is the results will be the same. However, in sex chromosomes, what we are going to have are different results. And then we have a back cross. A back cross. So a back cross is the hybridization of F1 progeny with one of its parents. So we are going to cross F1 offspring with one of its parent. So what does that mean? We have, remember we have the parental, the P generation, and we have the F1 generation. So if we cross F1 with a dominant parent, we see that all the progenes that will be produced will be dominant. Remember, the dominant allele is going to express itself whether in the presence of a recessive allele or, of course, in the presence of its same allele. So if the F1 generation is crossed with dominant parent, or the progeny or the F2 generation will actually be dominant as well. If F1 is crossed with the recessive parent, the individuals with both phenotypes are going to appear in equal proportion. So if F1 is crossed with a recessive parent, the result is that both phenotypes are going to appear in equal proportion. So the test of progenes produced during a back cross is what is known as a back cross ratio. The back cross ratio. The ratio of progenes that are produced during a back cross. And then we have what is known as a test cross. What is known as a test cross. So a test cross is a type, it's actually a type of back cross. A test cross is a type of back cross in which the F progeny is crossed with its double recessive parent. So in a test cross, the F progeny, the F1 progeny, is crossed with its double recessive parent. Remember, we have the parent here, which is recessive, and we have the dominant parent dominance there. So we have homozygous dominant, and we have homozygous recessive. So in a test cross, we're going to do a back cross where the F1 generation, the result of the F1 generation is crossed with the double recessive parent. So we're going to cross F1 generation with the double recessive parent. So the test cross is used to determine whether the individuals exhibiting the dominant character are homozygous or heterozygous. So a test cross is used to determine whether the individuals of the filial 1 generation, of F1 generation, that are exhibiting the dominant character are actually homozygous or heterozygous. So they're going to determine the genotype of F1. So a test cross is performed to detect the genotype of the F1 progeny, the F1 progeny. Yeah, so having said that, we've come to the end of our introduction to genetics, concepts and terminologies that we encounter in genetics. So in our next class, we are going to look at genetics, and we're going to look at classical Mendelian genetics. We're going to look at Mendelian